Now I want to introduce myself. Michael Gupta from Kathmandu Medical College, first year MBBS students. Now I am going to talking about the very important topic for the university examination, and that topic belongs from the pathology, that is apoptosis. Simply we have to know apoptosis means suicide of cells. Or death of cells. Means why the cell undergoes death. Why the cell wanted to take suicide. There is basically two reasons. One is if the cell has already done its function. Other one is if its DNA content is damaged. So the cell wanted to take suicide, wanted to commit suicide. At first, then how the cell undergoes the death or suicide? Simply we have to understood, like suppose this is a cell and this is central, that is where contains the nucleus, and this is the cytoplasmic content. Suppose the external stimuli, that is stimulus, which activates the enzyme present in the cytoplasm. That important enzyme is caspase. When the caspase enzyme is get activated, then it converts into, this cell is converted into many small cell. This small cell is called apoptotic bodies. Here in apoptotic bodies, cytoplasm is retained. Cytoplasm, nucleus is retained. Plasma membrane is intact, means not disrupted. This apoptotic bodies is engulfed by the phagocytes, by the phagocytosis process. In this way, the cell undergoes death. This is the mechanism actually. Simply we have to understand that when the stimulus is coming from the it may be outside or inside, which activates the caspase enzyme present in the cytoplasm. that caspase enzyme activation causes the splitting of into apoptotic bodies where the cytoplasm nucleus is retained, plasma membrane is intact. Now we have to understood the more and more concept of apoptosis. This is also defined as genetically programmed cell death. Now we have to study about the types of apoptosis. That is physiological and another one is pathological. Example of physiological, the important example is embryogenesis. Like in embryonic condition, the hands is like this, but the apoptosis occurs in it, which converts into the phalanges and digits. And the important example of pathological apoptosis is in the AIDS, during AIDS, in AIDS condition, during the AIDS, there is depletion of CD4 plus T cells. Now the other concept of apoptosis is when actually in apoptosis there is no cytoplasmic leakage. There is no cytoplasmic leakage. So no inflammation is seen during the apoptosis, which is really very important concept for the MCQ. Now I am going to talking about the mechanism of apoptosis. There is two pathways of apoptosis. One is extrinsic pathway another one is intrinsic pathway. In extrinsic pathway, the word itself says that the stimulus is coming from the outside, which activates the caspase enzyme, and then finally the apoptosis occur in that cell. So we know in the normal cell, this is a cell, this is the this is the nucleus, This is the cytoplasm. This is the mitochondria. This is the endoplasmic reticulum. And this is the lysosomes, and this is the ribosomes. In extrinsic and intrinsic pathway, this is the mechanism. In extrinsic and intrinsic pathway, there is extrinsic pathways and intrinsic pathways. Both is divided into two types. That one is initiation and another is execution. In a similar way, intrinsic is also divided into initiation and execution. Now I am going to talking about the initiation steps of the extrinsic pathway. Extrinsic pathway. Means stimulus is coming from outward. Suppose this is the normal cell, and in this cell there is the receptor. Which is called death receptor. This is actually a protein, and death receptor is of two types. Death receptor may be Phage or CD95 receptor. This is the death receptor which acts as in that site the ligand is binded. Suppose here is death receptor, that is phage, then the phage ligand. This is the what? Phage ligand. When this phage ligand bind with this receptor, phage bind with this death receptor, then these all death receptors are coming together in the cytoplasm. All death receptors are coming together in the cytoplasm, forming FADD. This FADD is phage associated death domain. This phase associated death domain now activates the procaspase 8 to caspase 8 in the cytoplasm. This is inactive form and this is the active form. Means our main target in apoptosis is to activate the caspase. We look earlier that when the caspase get activated, then apoptosis occur. So our main target is how the caspase get activated. Once I repeat it, in normal cell there is in the cell membrane there is the death receptor in which the phage ligand is bind. So all the death receptors are coming together forming the phage associated death domain, which activates the procaspase 8 enzymes to caspase 8 in the cytoplasm. Here procaspase 8 is present in inactive form in the cytoplasm, which get activated by the FADD into caspase 8. This is the extrinsic phase of apoptosis. In extrinsic phase, this is called initiation phase of the extrinsic pathway. Now I am going to talking about the initiation phase of the intrinsic factor. Which is really very important and little bit difficult, but I make it easy. So look it very carefully. Now I am going to talking about the initiation phase of intrinsic pathway. Means stimulus is coming from the inside. Suppose this is the cell in which this is the mitochondria and this is the DNA content. As you know, apoptosis only occur if there, and I am already told you that in two conditions there is cell decided to take suicide. One is when their function is already done and not in use, and another is when their DNA content is damaged. So now I am talking about here DNA damage in the initiation phase of intrinsic pathway. This is the mitochondria. Between the two membrane of mitochondria there is a protein. That protein is cytochrome C. Very important protein. This is the cytoplasm. Normally, these cytochrome C cannot enter into the cytoplasm because there is the protein there is which is called anti-apoptotic proteins, that is BCL2 and BCLXL, BCL2 and BCLXL, which acts as guard, and this is also called as anti-apoptotic molecules because these can prevent the exit of cytochrome C into the cytoplasm. But when the DNA gets damaged, it sends signal to the mitochondria. Then these anti-apoptotic molecules get replaced by the apoptotic molecules. That is BAK, BAX, BIM. These are the apoptotic molecules. Means apoptotic name itself suggests that this might cause what? Apoptosis. Now, when the anti-apoptotic molecules get replaced by the apoptotic molecules, then the only cytoplasm, cytochrome C, is coming to the cytoplasm. And as you know, in cytoplasm there is the number of caspase enzyme. Caspase, suppose caspase 9, which is binds with APAF1 in the cytoplasm. But when the cytochrome C is coming into the cytoplasm, cytochrome C break the bonds between the caspase 9 and APF1 and moves with the APF1. Then caspase 9 get free in cytoplasm. Which get activated. Means in initiation phase of extrinsic pathway we activate the caspase 8, and now in initiation phase of intrinsic pathway we activated the caspase 9. Means the two enzymes get activated. Now the execution phase of both intrinsic and extrinsic pathway. The execution phase of both extrinsic and intrinsic pathway is same. Look, intrinsic pathway and extrinsic pathway, we activate the caspase 9. In intrinsic pathway we activate caspase 8. Then these caspase 9 and 8 can finally activates the caspase 3 and 7. Then after the activation of 3 and 7, all the remaining caspase get activated. And when all the caspase get activated in the cytoplasm, which leads to cell death. Very easy. cell death, and we already saw that in this way the cell is converted into apoptotic bodies. And now the very important question is that how the phagocyte identify that this is the apoptotic bodies and we have to engulf it, and this is the normal cells and we have not to engulf it. The main reason is now, so you are going to talking about the what mechanism of phagocytosis. Very, very important. In normal cells, there is the phosphatidyl serine. This is the lipid, that is phosphatidyl serine is a lipid which is generally present inside the cell surface in normal cell. But in apoptotic cell, these phosphatidyl serine comes outside. This is phosphatidyl serine. This is phosphatidyl serine. So the apoptotic bodies is get engulfed by phagocytes because there is phosphatidyl serine, that is, this is the phosphatidyl serine which is outside the cell surface. So the phagocytes identify that this is the apoptotic bodies and we have to engulf it. This is the mechanism of phagocytosis. Why the apoptotic bodies undergoes the phagocytosis and how. Apoptotic bodies can undergo phagocytosis because of phosphatidyl serine in their outer surface. Now very important topic is morphological changes occurs in apoptosis. That is morphological changes in apoptosis. Morphological changes occur in apoptosis. This is the normal cell. This is the cytoplasm. Normal cell. Suppose this is a step first. Then after the cell shrinkage occurs. So cytoplasm also get shrunken. Second, this is cell shrinkage and cytoplasm and all the cell organelles comes together, so appear as pink, called as eosinophilic. In third step, there is the nuclear condensation. There is what? Nuclear condensation. Nuclear condensation. This condensed nuclear is called pycnosis. And fourth step is breaking down of that nucleus into the fragment. This is called karyorexis. And fifth step is the formation of blebs. Simply we have to know blebs means the plasma membrane is not disrupted, means it remain as, means plasma membrane is intact, not leakage of cytoplasm. Simply we have to understood like that. Then after, blebs convert into apoptotic bodies, small apoptotic bodies. And as we know earlier, apoptotic bodies, cytoplasm is retained, nucleus is retained. Then this is the what? Apoptotic bodies. Now the phagocyte came and saw the lipid in the outside the cell surface of apoptotic bodies and engulf it. So the sixth stage and the seventh stage is phagocytosis. This is the morphological changes occurs in the what? Apoptosis. Now I am going to talking about the very important MCQ may ask in the PG entrance or our university examination from the morphology of apoptosis. My question is that what is the earliest features of apoptosis? Then earliest features is what? Cell shrinkage. Very important. Then other important question, very important, that is what is the pathological feature of apoptosis, and that one is pycnosis. What is pycnosis? Actually nuclear condensation. We have to remember this MCQ for the university examination. What is the earliest feature of apoptosis? That is cell shrinkage. And what is the pathological feature of apoptosis? That is pycnosis, means nuclear condensation. Now I am going to talking about the diagnosis, how to diagnose that this is apoptotic bodies. Diagnosis of apoptosis, very important for your information. One is apoptosis marker. Apoptosis marker, that is annexin. Annexin 5 is called apoptosis marker. This is actually a liquid form. Is a liquid form. This is a what? Liquid form. These can identify the apoptotic bodies. So annexin 5 is considered as apoptosis marker. Now the video is ended. So thanks for watching this video.